Today we are going to discuss about one of the main important biomolecule, that is the carbohydrates. Before moving toward the carbohydrates, that is a biomolecule, we need to tell about the biomolecules. What is biomolecules? The molecules present biologically that are vital for our system or living system, they are known as the biomolecules. There are different type of biomolecules. They are carbohydrates, proteins, lipids and nucleic acids. And one of the most abundant and the most important biomolecule is carbohydrate. You will already know. You already will be knowing about the energy molecule, that is the glucose of our system as well as all living system, that is the glucose. So that is one of the most important biomolecules that take place in the carbohydrates. Now coming to the carbohydrates, they are the most abundant biomolecules and they are known as the polyhydroxyaldehyde or polyhydroxyketones. Polyhydroxy, that means there are large number of hydroxyl group in their body as well as aldehyde group, and the polyhydroxyketone means there are large amount of hydroxyl group with keto group, and they serve as the major source of energy for the living organism. We all are living organism. This living organism is having an energy that is from the carbohydrates. Now coming to their function, we already discussed about the first function of carbohydrate is energy, that is they can act as source of energy. Coming to the source of energy, ATP, we already know that ATP is a high energy molecule that is required for our system, that is adenosine triphosphate, and this energy molecule is come in our body through glucose, that is a carbohydrate. Glucose, after we are taking food material, that includes the carbohydrate, and the basic component of carbohydrate is glucose, and whenever this glucose is entered into our system, especially in blood, this glucose is converted to ATP through metabolic pathway that we will further study afterwards. And that is thus one of the main function of carbohydrate is they can serve as source of energy in the form of glucose. That means this glucose can be converted to energy molecule, that is the ATP. And the second one, that is structural component, that is the carbohydrates can act as one of the most important structural component in systems such as prawns, crustaceans, etc. In their shell wall, we can see a molecule known as a chitin, and this chitin is a carbohydrate molecule that is providing the structural property to that particular organism. And one of the most important example or most known example is the cellulose. That particular cellulose is mainly found in the plant cell wall, and that cellulose gives structural property to that plant cell wall. Plant is having a particular structure through the cell wall, and this cell wall is made up of the cellulose, that is a structure that provides structural components to that particular plant cell wall. Therefore, that is a second important function of the carbohydrate. Coming to the third one, they can serve as a storage form of energy. That means glycogen. Glycogen, we all know glycogen is a carbohydrate. It is a polymer of glucose, and this glycogen is a storage compound found in our body, as well as starch. That starch is a storage plant storage molecule, and glycogen is the animal storage molecule. Both the starch and glycogen are carbohydrates, and this can serve as a reserve food whenever we need energy, or in case of a starvation time we are not able to have an energy. At that particular time, this glycogen can be converted to glucose for releasing the energy as ATP energy molecule. Therefore, the third function is storage. And coming to the fourth function, they can act as the building macromolecules such as RNA and DNA. We all know DNA, that is a nucleic acid, that is a deoxyribose sugar. In case of DNA, ribose, that is a carbohydrate molecule found in the nucleic acid that can serve as the building that ribose is a carbohydrate that can serve as building molecule of nucleic acid, that is the other function. Coming to the last function, carbohydrate can participate in different type of biological transport such as active transport, passive transport, etc., down membrane equilibrium, etc. Then cell to cell recognition. These are all the other functions of carbohydrate. Actually, there are different varieties of functions for carbohydrate. Here we represent the most important function. The first one we already told that they act as source of energy. Second one, they can serve as a structural component. Third one, they can serve as storage molecule, storage energy molecule. And fourth one, they are the building molecule. And last, they can participate in different processes such as inflammation, body's biological transport, cell-to-cell recognition, etc. These are all the functions of a carbohydrate. Coming to the carbohydrate structure, we already know that simple carbohydrates are known as saccharides or sugars. We call the simple carbohydrates as sugars or saccharides, and some of the most important simple sugar you will be already know about some of the examples. But these are some of the examples of carbohydrate. That is, first one is the glucose. Then that glucose we already know that is the sugar present in our blood. Second one is the fructose, especially in fruits and in honey in case they are the sucrose. And lactose, that is the milk sugar. The sugar present in milk is lactose. Maltose, malt sugar, especially in sugars, malt, etc. We can see maltose. And starch, that is a polysaccharide, that is storage form in plants. Plants store their food in the form of starch, and the starch is formed from the photosynthesis. And cellulose, that is the sugar in the plant saliva that gives a structure that we have already discussed, that sugar cellulose is structural property to the plants. And last one, that is the glycogen that give the storage component or the storage of sugar, etc. These are all the predominant or the most commonly sugars found in our system, as well as not in particular in our system, around system, that is glucose, fructose, lactose, maltose, etc. Coming to the coming to their reaction, so coming to their form that is found in our system or in our living system, we can say that carbohydrate form is bound form rather than free form. For example, we already told about the glucose, etc. Coming to the glucose, etc., we can see they are found in bound form. In case of the lipid bilayer, we already know our cell, all organism cell is made up of cell membrane, and cell membrane we can see carbohydrate. The carbohydrate is in bound form, that is they are bounded with the protein for the formation of the glycoproteins. Polysaccharide, we already discussed such as starch, cellulose, chitin. These are all the polysaccharides that are found in the bound form rather than the free form. Nucleic acid, that is the building molecule, that is deoxyribonucleic acid, where that particular sugars get bind with the nucleic, etc., mucopolysaccharide, etc. That is why the carbohydrate will be found bound form rather than the free form. Coming to the classification of carbohydrates, carbohydrates are classified into simple carbohydrates or monosaccharides, oligosaccharides, and polysaccharides. These are the three classes of carbohydrate. They are monosaccharides, oligosaccharides, and polysaccharide. At first, from the name mono, that means simple sugar. Monosaccharides are known as simple sugars. Some of the examples are glucose, fructose, and galactose, etc. And second classes, that is the oligosaccharide, up to 10 molecules. Up to 10 molecules is known as of 10 to 20 molecules. They are known as oligosaccharide. Specification up to 10. In certain books, they are depicting as 20. That is why we are told about 20 molecule. Now we can tell that the oligosaccharides are the second classes where we can see up to 10 molecules of sugars. In that particular oligosaccharide, we can see 2 molecules of sugars known as disaccharides. If there are 3 molecules of sugar, they are known as trisaccharide. If there are 4 molecules of sugars, they are known as tetrasaccharide. Up to octa, that is 9 molecules. That goes on. And last classes, that is known as a polysaccharide. The number of sugars greater than 10 to infinity, they are known as the polysaccharides. And again, these polysaccharides are classified into two. They are homopolysaccharides and heteropolysaccharide. Here, homo means the sugar units combined will be the same. That is homopolysaccharides. And in case of heteropolysaccharide means the sugars that is combined will be different. And certain cases we can see the complex carbohydrates in polysaccharide. Complex means whenever that particular carbohydrate is binding with other molecules such as proteins, lipids, etc. For example, glycolipid, that is a combination of carbohydrates and lipids. Glycoprotein, that is a combination of carbohydrates and proteins, etc. Now we are going to discuss in detail about these all classifications with structure. At first one, we already told that monosaccharide, they are known as simple sugars. And again, these monosaccharides are classified depending upon the different type of criteria. And the first one, that is the number of carbon atom present in the molecule. That is, carbohydrates are classified into three. They are mono, di and poly. Again, the monosaccharides are classified into two based on the number of the carbon atom present in their system and whether the aldoses or ketosis. And first one that is number of carbon atom present in the molecules. They are classified into mono, aldoses, etc. That means colorless, crystalline, etc. These are all the examples of the monosaccharide. Here in the first figure, we can see only this is the simplest sugars, that is glyceraldehyde and dihydroxyacetone. Here we can see the aldehyde group and here we can see the keto group, but the number of molecules of carbon is same. That is, here we can see 3 carbon and here also 3 carbon, but only difference is in the functional group. Here the dihydroxyacetone we can see a keto group, whereas in the glyceraldehyde we can see a aldehyde group. That is the examples. This classes comes in trioses, that is number of carbon is 3, they are known as a trioses. Now here the example is 1, 2, 3, 4, that is a pentasaccharide, that is 4 molecules of sugars. Here also this is the aldehyde group, here is also the keto group. Therefore, the number of, depending upon the number of carbon atom as well as depending upon the ketosis or aldoses, that is about the functional group they are classified. That is all about the monosaccharides. Coming to their property, they are colorless, crystalline, solid. They can have 2 or more hydroxyl group, and the simplest monosaccharides are the 2,3 carbon, that is known as the glyceraldehyde and dihydroxyacetone. Coming to their structure, here we can see the glyceraldehyde and dihydroxyacetone. In this structure, we can see 3 carbon molecule. Here we can see the aldehyde group. In case of dihydroxyacetone, this aldehyde group is removed by the keto group. And these are all the examples. This is the simplest monosaccharide that is found in the trioses with 3 carbon atom. The examples are glyceraldehyde and dihydroxyacetone. This is all about the monosaccharide. Coming to oligosaccharides, we already told that oligosaccharides contain up to 10 molecules of sugars. We can also define these oligosaccharides as the compounds which upon hydrolyze yield 2 to 10 molecules of sugars. That particular type of sugars are known as oligosaccharides. And whenever that upon hydrolysis, this oligosaccharide yield 2 molecules of sugars, they are known as disaccharide. Whenever they yield 3 molecules, they are known as trisaccharide, up to 10 molecules. For example, here disaccharide means upon hydrolysis of the oligosaccharide leads to the formation of 2 molecules of sugars, and their examples are sucrose, lactose, maltose, etc. And in case of trisaccharide, there is one example is raffinose and then geniose, etc. Now we are going to discuss about the disaccharide, that is classes that comes in the oligosaccharide which upon hydrolyze the 2 molecule. And each disaccharide has a specific glycosidic linkage. That means whenever the 2 sugars combine together for the formation of sugar units, that linkage or that bond is known as the glycosidic bond. And 3 most common sugars that is found in the oligosaccharides are sucrose, maltose and lactose. And these are all the examples: maltose, lactose and sucrose. Here coming to their structure, we can see the maltose upon hydrolyze yield 2 molecule of glucose, and lactose upon hydrolyze yield 1 molecule of glucose and 1 molecule of galactose, and at last it's sucrose which upon hydrolyze yield glucose and fructose. Therefore, we can tell that the particular maltose is formed by the combination of 2 molecule of glucose, whereas sucrose is formed by the 2 combination of glucose and fructose, and at last the lactose is formed by the combination of glucose and galactose. Now we are going to discuss about each in detail. At first, sucrose. We all know sucrose is a sugar that is found in fruits, honey, etc. They are also known as a table sugar, and it is the most abundant disaccharide. comparing with the other disaccharide, we can say that sucrose is the most abundant disaccharide. Coming to their structure, here we can see they are formed by 1,2 glycosidic bond. That means the first positions of glucose and second positions of fructose bond is between that two positions. That is the glucose first position and fructose second position, alpha 1,2 glycosidic bond, and it is a non-reducing sugar. Sucrose is a non-reducing sugar. That means there are two types of sugar that we have already studied, that is non-reducing and reducing depending upon their reducing property. Sucrose is a non-reducing sugar because their alpha carbon atom comes in the bond formation. They can never rotate. That is why sucrose is a non-reducing sugar. Next one is maltose. Maltose is formed by the combination of two glucose molecules. Here the linkage is 1,4 glycosidic linkage. 1,4 glycosidic linkage is 1 and 4. That is first position of glucose and fourth position of other glucose molecule combined to the formation of maltose. And coming to the lactose, that is the sugar found in the milk that is formed by the combination of glucose and galactose. Here also the bond formation is 1 and 4. 1 and 4, that is galactose first position and glucose fourth position. Bond is formed between this position, and lactose is sweeter and more soluble than ordinary lactose. That is, we all know there are alpha and beta form. This beta lactose is more sweeter comparing with the alpha, and upon hydrolysis it will lactose by the enzyme lactase. This is all about the oligosaccharide. Coming to the polysaccharide, upon hydrolysis it will yield more than 10 molecule of polysaccharide. They are more than 10 molecule of sugars. They are known as the polysaccharides. And these polysaccharides are again classified into homopolysaccharides and heteropolysaccharide. Examples, this is already mentioned earlier. Now we can move to each polysaccharide. These are all the properties of the polysaccharide that we have already discussed. Up to 100 to several number of more sugars combined together for the formation of the polysaccharide. And the biologically important polysaccharides are starch, glycogen and cellulose. We need to go through that particular polysaccharide. The first one is starch. Starch is a combination of amylose and amylopectin, and starch contains a mixture of both amylose and amylopectin. Amylose means the bond formation between alpha 1,4, though we can never see branches, but in case of amylopectin we can see branches, and that particular type of amylopectin is formed by alpha 1,6 glycosidic bond. Therefore, in case of starch we can see two bonds there, alpha 1,4 glycosidic bond and alpha 1,6 glycosidic bond. Amylose is unbranched, whereas amylopectin is branched. We already mentioned that amylose is branched, is unbranched, and it is linked by alpha 1,4 glycosidic bond. Amylopectin by alpha 1,6 glycosidic bond. This is the structure of the starch. Here we can see this is amylose that is having straight chain, and the amylopectin that is formed by the linkage, that is alpha 1,6 linkage, gives the branches between the bonds. And this is all about the starch. Now coming to the glycogen, this is the most other important polysaccharide found in our system. We already told that glycogen is a storage compound. This is also formed by the glucose units, that is a glycogen, and starch is almost similar in structure, whereas the glycogen is a storage molecule. It is a white powder. Whenever we get starved or animals get starved, this glycogen is broken down for the formation of the glucose, and it gives energy to that particular system. Here also 1,4 linkage is formed in case it is same as that of starch. Coming to the cellulose, and the cellulose is a structural polysaccharide, whereas starch and glycogen we already told that they are the storage polysaccharide, and in case of cellulose it is structural polysaccharide. Here the main difference is the glucose found in cellulose is beta glucose, whereas in starch and glycogen it is all about the alpha glucose. Here the bond formation is beta. Here the glucose molecule is beta glucose. That is one of the main Characters or main specifications of the cellulose. Some animals contain cellulose enzymes in their body to distract or for digestion. For example, in cows, herbivores, they are able to utilize plants because in their bodies, in their organisms, in the same, there are certain microorganisms that are having the ability to produce the enzyme cellulase. Cellulase is an enzyme that will break down the cellulose. Therefore, they can easily digest cellulose, but in our case, and as in our system, they are not able to digest because we lack cellulase enzyme. This is all about the cellulose. Now coming to the chitin. Chitin is another structural component mainly found in crustaceans, cockroaches, etc. It is also structural component. Here the units sugars in the chitin is a modified sugar, that is an acetyl galactosamine. And it is a modified sugar. Here we can see in the second position the modification occurs, the glucose molecules. That is a modified sugar is found in the chitin. That modified sugars combine together for the formation of the chitin. And chitin has several applications, such as they can be used as surgical threads, as it is biodegradable. It is widely used. This is all about the chitin. Therefore, today we have discussed about the carbohydrates, their properties, their classification, and their application. And this is all about the carbohydrates. Thank you.